Well, ladies and gentlemen, let me introduce you to our next speaker. Well, Mr. Pranav Tiwari has over 20 years of experience in building large-scale products and running engineering organizations. Before joining Cellworks, he was responsible for managing Google India Engineering in Bangalore. Ladies and gentlemen, please put your hands together and welcome Mr. Pranav on stage.
Can you all hear me? Good. So let me start by asking how many people here are doctors or medical professionals? I didn't expect any hands to go up. How many of us are engineers or computer scientists? I guess most of us. That's like me. So I started venturing into this field a couple of years back, and many reasons for that, but I found it particularly fascinating. And I hope that some of the glimpse that I give you introduces you to what drew me here. So what has happened is, as computer scientists or software engineers, each time our field makes a stride in solving a problem or achieving something big, I derive a lot of vicarious pleasure from that. It's like a triumph for me. So when Google DeepMind went ahead and beat Lee Sedol, there was a little silent yay that I uttered. I'm sure many of you felt the same. But if I start thinking about what software engineering, not all engineering, but software engineering has contributed to the field of medicine, I start drawing a blank. Yes, we have done a little bit here and a little bit there. We've written software to control the instrumentations and machines that do various things, but fundamentally, we have not changed anything. 50 years later, we'll not be thinking of hospital management systems as the biggest achievement that transformed medical science. Yes, they've made it more efficient to run hospitals, more efficient to utilize the resources there. But you would not think of that 50 years ago, 50 years later, that's an achievement that our field should be proud of. What I'm going to talk about is how this is changing now and why this is a time where medical science and software engineering are starting to get a lot more intertwined. And some of that is due to availability of a lot of data. Some of that is due to availability of a lot of measurement techniques which are giving us precise results, but which are very hard to interpret manually. All these reasons are combining to bring these two fields together. And that's what my talk is going to be about. So in this talk, I'm going to talk a little bit about what diseases and therapies have been historically, how our response to diseases has been, what advances we have made against it, and where we still struggle. In the process, I'm going to talk about cancer, which is a particularly difficult subject. It's a particularly challenging disease, both in terms of the amount of effort, the amount of money we spend as a society, and the impact it has on the patients, their families, and everybody else. The reason I bring up cancer is that this is one of the places where I see a large opportunity for fusion of our software engineering techniques along with medical therapy. And finally, I'm going to go on to talk about why personalized medicine makes a lot of sense for something like cancer and what we have done in the space of personalized medicine in trying to treat cancer or trying to address the challenge of cancer. So the landscape of diseases has changed quite dramatically over the last 2 centuries. A couple of 100 years ago, we had a very different set of diseases that were like the scourge of mankind. In 1900s, I'm not sure if you can read this, but I'll read the top 3 for the benefit of those who are sitting a little far. So in 1900s, the top 3 killers used to be pneumonia, TB, and the stomach bug, diarrhea and enteritis and so on. So these 3 together used to claim about 45% of all lives. Now that's huge. 45% of people falling to pneumonia, tuberculosis, and gastroenteritis or diarrhea. This was the biggest medical threat people used to see at that time, and our hands were tied until we started discovering ways to understand what's causing these. That happened over the course of 1900s, and by the end of the century, the landscape has dramatically changed. By the end of the century, we still lose people to pneumonia, but the numbers have come down so dramatically that it doesn't figure in top 3 anymore. The biggest 3 killers today happen to be heart disease, cancer, and stroke. Stroke is another form of some malfunction in the blood flow, which restricts blood flow to the brain. But comparing the top 3 from 1900 to the top 3 in 2000, you can clearly see that we are looking at a completely different set of diseases, and we are looking at a very different challenge in terms of medical science. This has happened a lot because of our victories that we have happened to achieve over the infectious diseases like pneumonia, tuberculosis, and so on. These diseases over the course of this century, we discovered what causes these diseases. We started looking for ways to address them and eventually managed to conquer them. Many of the diseases like smallpox and polio, they've been eradicated from large parts of the world where they used to be extremely deadly at one point of time. It's not so anymore. So the landscape has changed, and what has changed along with that is that our view of these diseases also has changed quite dramatically. The way we used to think of diseases, what is challenging, what hurts us is very different now. And along with that, we need to change the way we treat medicine and treat therapies. So I divide diseases in two classes. Think of infectious diseases on one side and the non-communicable ones on the other side. And this is a very broad classification, but it becomes important in terms of how we approach our therapies. Infectious diseases, all of you know, are caused by viruses, bacteria, things that our body contacts from the outside, external environment. And that's pretty convenient because once our body has contacted something from the external environment, it also gives a therapist, a medical therapist or a medical practitioner, a target. It gives the pharma companies a target to develop a drug or a molecule that hurts the external organism but is non-toxic to our body. That's the only challenge that remains. It's a big challenge, but that's the only challenge that remains. So what it means is the moment I find that molecule, it doesn't matter who has that disease, I can give the same drug molecule pretty much to every individual, and we'll have the same results. We'll be able to kill the external organism, we'll be able to restore the body's regular functions, and we'll be able to have a healthy individual. And that's pretty much what worked in our favor as we dealt with infectious diseases. We were able to find ways to prevent them, we were able to find ways to build immunity against them, and we were able to find these universal solutions where a single therapy would work against that disease. It doesn't matter who has that disease. Now that was fantastic, and that allowed us to go from being completely focused on diseases like pneumonia and so on to not think of them as too much at all. Now, all these advances happened during last century, but during this period, we did not have any strong achievement, any large achievement that helps us against the non-communicable diseases. Now, these non-communicable diseases are not caused by a single organism. There is no single organism that's causing my heart disease or somebody else's heart disease. It's a malfunction emanating from within my body. There's no single reason that gives one person a cancer, because the reason that leads to cancer in one person is very different from the reason that leads to cancer in another person. So we don't have a single treatment. We can mitigate the symptoms. We can try to reduce the chances of that disease from having an onset, but that's all we can do. We cannot cure these diseases in the same way that we cured influenza or pneumonia. We don't have a single pill that everybody can take and the disease will be cured. That's a problem, because our approach that worked so effectively against infectious diseases of taking one pill and that will address all problems for everybody, or address that problem for everybody in the world, that approach completely fails. So we don't have universal treatments. We have some preventive measures. We can try to take care of things that reduce the likelihood of onset of a certain type of disease. However, we end up treating largely symptoms rather than the root cause itself. So here's the story so far. In the last century, we made quite a lot of advances in our fight against infectious diseases. We have a lot of that has been rooted in scientific advances like ability to conduct surgeries with anesthesia, broad spectrum antibiotics, ways of preventing it by either sanitation or sterilization of surfaces we get in contact with, or by vaccination, or the development of all the pathology in which we are able to detect what's going on much better than what we were able to. So each of these has contributed, and we've managed to largely be in control of infectious diseases. Yes, new infections keep cropping up, but we have a general direction in which we approach these infections, and we have a general way of solving these things. That gives a little bit of comfort that, yes, when Ebola strikes somewhere, at some point we'll either develop a vaccine against Ebola, or we'll find a molecule that attacks the Ebola virus and leaves the rest of the body intact. On the other hand, on non-communicable diseases, or NCDs as I refer to them, these have become a large challenge. We don't have any hope of achieving a single universal treatment that works on everybody. That basically means that each time an individual comes to a doctor's office, the doctor is prescribing a medicine to him, but that medicine is not necessarily going to address the disease in the same fashion as the previous patient. The response of that human being's body to this medicine, or the response of that disease is quite varied. So I'm in particular interested in cancer. Two reasons. It affects about 25% of the people, 25% people in the world. In some genetic groups, in some ethnic groups, it's much larger than 25%. And it's growing as we are able to handle other diseases. This is one of the diseases, I mean, just by a process of elimination, the likelihood of this becoming more prevalent, or the likelihood of me succumbing to cancer, is much larger because I'm not going to succumb to smallpox. So the reason, so the importance of this keeps growing as we are able to claim victories on other places. I found that pretty staggering to learn that about 1 in 3 people who are alive today are likely to contract cancer through their lifetime. And the survivability remains still fairly, I would say, compared to most other diseases, fairly low. And all these factors make it an extremely challenging beast to deal with. The total cost that we incur as a society is staggering. There's a huge amount of money that goes in taking care of people who are ill with cancer, both in terms, and many of the medications are extremely expensive. Then the disease drags on for a while, even though there is recovery, there is always a likelihood of recurrence. So each of these things keep making this disease extremely difficult to deal with. So let's talk a little bit about cancer. I mean, so cancer happens to be a disease of the cell. Cell, as we all know, is the basic building block of life. All of us are basically comprised of cells. Some, I believe, 30 trillion cells in our human body. Inside the cell, we have what we call DNA. DNA is basically a code for life. It encodes how this cell is going to behave, what the rate of its reproduction is going to be, how it's going to, what its lifespan will be, how much energy it should consume, whether that cell remains anchored to one point, whether it moves. Each of these aspects are encoded or controlled by our genes, which are essentially embedded in the DNA. Now, a human body has roughly 25,000 genes. Now we know that from the Human Genome Project. And these genes are responsible for creating chemical molecules, proteins, which eventually control the behavior. Now, the proteins are responsible for listening to external communication for the cell, altering the behavior of the cell, making the cell move, die, reproduce. Each of these aspects are controlled by the protein. So this one. Okay, so this pointer is not going to work. But this one thing that I write about genes leading to proteins, which leads to function, is something that I want all of us to internalize, because this is what is going to keep coming back to this talk. Now, the cell functions include many, many things, including the growth of the cell, division of the cell, metabolism, its self-destruction. Each of these aspects are controlled by the proteins that are present in the cell. And these proteins, as we know, are regulated by the genes. In a normal cell, these functions have a very intricate balance. If a cell, for example, refuses to die, that's a problem because other cells are coming in place. So if the cell doesn't die and this type of cell doesn't die, then we have an accumulation of cells. That's never a good thing. If the cell is not able to divide fast enough and replicate, for example, if my blood cells don't replicate fast enough, then I start getting blood deficiency. So each of these functions, they have a balance, and that balance needs to be maintained for the organism to live in a healthy way. What happens in cancer is that some of these functions, they don't function naturally. They don't function in the natural harmony. This harmony is disrupted, and as a result, you have some growth, some aspect of misbehavior of these cells. One type of misbehavior could be that the cells start dying, which is not as bad because once that type of cell dies, other healthy cells take its place. But what happens if the cell starts growing indefinitely? Now, there's no way to for the other cells to take care of that, and that's what results in a tumor. That's what results in a cancer. So cancer comes from uncontrolled growth of the cells. These cells keep dividing repeatedly, even when not needed, and these cells refuse to die. When a cell gets into that combination, you eventually start getting accumulation of these cells, and over time, you end up seeing symptoms of cancer. It's a very slow process. I was mistaken before I started reading about this. One of the mistakes I had assumed based on popular perception was cancer is a very rapidly growing scourge. It suddenly takes over your body. It doesn't. A cancer typically takes about 15 to 20 years in a human body before it starts showing any symptoms. It takes about 15 years before it can be detected inside a human body. It takes about 1 billion cells of a billion cancerous cells in our human body before we are able to observe any effects of that on the body. So by the time we find that the cancer has taken root, it's already got about 1 billion cells to hurt us. So what happens is, on the left side, you have a picture where, let me just throw a term called tumor suppressor gene. A tumor suppressor gene is something that limits how rapidly a cell can divide. It produces a protein which inhibits the growth or inhibits the desire of a cell or inhibits the likelihood of that the cell will divide. So when in a normal system, there is a certain concentration of that protein needed so that the rate of growth of that cell can be controlled. But there are times when this gene does not function properly. There are times that this gene starts misbehaving. It doesn't produce the right kind of protein, or the protein that it produces is not adequate. As a result, whatever was regulating the growth or regulating the reproduction of the cell goes away. So if the cell was reproducing at a rate R before, that R becomes larger. The sad part is these genes are copied down when the cell divides. So a malfunctioning gene in one cell would be replicated as a malfunctioning gene in both the children that it produces. So both the children now produce divide at a larger rate, and their children, and their children, and so you know what happens. So I want to think of this as cancer. Cancer is a function of incorrect or an imbalance in the functions that the cell is involved in. Now, this used to be the diagram of the cell I learned in school. Basically, it's a biological diagram where all the organelles or organs of the cell are represented as a graphical form. Their functions are depicted qualitatively. And we know that mitochondria has a certain function, and the DNA has a certain function, and this and that. But all these functionalities are expressed fairly qualitatively. And that confuses people like us, because whenever something is expressed qualitatively like that, there's very little we can do about that. So until we can turn this into a quantitative model, it's very difficult to use computing to figure out what's going to happen to this cell. So I prefer a picture like this, where I wish, unfortunately, you can't see this on the screen, but so I prefer a picture like this in which the cell is represented not in terms of its organs or subcomponents, but more in terms of the molecules that are present in the cell and what they do. So this may be unreadable, but what it says is that this is a signal coming into the cell which says you are supposed to die. Now, how does this signal come into the cell? A signal is sent to the cell by the presence of some other molecules produced by some other neighboring cell. So this signal is received by a gene called FADD. FADD produces some protein that has an affinity to the shape of the protein that's coming from here, so they're able to bind together. When that signal comes to FADD, that triggers that signal to another gene called caspase-9, which eventually signals apostosis. Apostosis is the process of self-destructing itself. Now, from an external molecule, a signal can be sent to the cell, which leads to the cell's self-destruction. The concentration of FADD, the concentration of caspase-8, determine how effectively that signal can be communicated to the cell for its decision to self-destruct, which basically means that if there is any inhibition in production of FADD or inhibition in production of caspase-8, that would lead to the cell's propensity to self-destruct go away. Now, as you can see, the apostosis is not controlled only by caspase-8, but there are many other signaling pathways that are also coming to that process. So, and also notice that there are, if you look at this pathway that comes to JAK2, that's going to, on one side, that's going to apostosis. On the other side, it's going to cell proliferation. So we have each molecule controlling multiple functions, participating in multiple signaling pathways. Their rates can be studied, at what rate they will be controlling this pathway versus that pathway, at what concentration do they become active, what is the likelihood that if it receives a signal, will it actually lead to a cell proliferation or apostosis or not. We can study each of these aspects, and then we can start modeling this mathematically. That's the reason I like this model much more than, or this presentation of a cell much more than the previous picture I showed you, where you could tell me that a certain function is responsible for cell division, but how do I know how the cell will change if a mutation occurs? I cannot model that there. This is a model which I can comprehend as a mathematician or as a computer scientist, where we can start looking at this as a network of signals, as a communication network in which signals are being received from outside, they are being propagated through various function blocks within the cell, and then we can start computing if one of these paths is pinched by a little bit so that the signal flow is obstructed, what would happen? And if 200 of these paths are pinched by a various amount at various places, what would happen to this cell? And if I want to rectify that change that has happened, what should I do? Where should I intervene? We can start thinking about that using this model. So that's what we started looking at in our company. And we started modeling the cell not as a qualitative collection of components, but as a quantitative signaling network. So here is a cell. Let's think about how cancer develops in a cell. Here is a cell. I've shown what is on top in green as chromosomes. Chromosomes are, you don't need to worry about what they are. Basically, they are, think about them as a house for our genes, which is the encoding of the genetic material. These chromosomes are replicated every time a cell is divided. A cell is divided, you get multiple copies of, there are DNA strands that are inside the nucleus, and these DNA strands, each DNA strand replicates itself and becomes 2 DNA strands. The genes are embedded on top of the DNA strands. Think of a DNA strand as a sequence of alphabets. So there's a very large sequence of alphabets that encodes what the cell is supposed to do. Every time the cell divides, you take this sequence of alphabets, make another copy of it. Each cell inherits one of these 2 copies. Now, there are 25,000 genes in each cell, as I said earlier. Average length of each gene is about 10 to 15,000 alphabets. We call them bases. So each gene is of the length 10 to 15,000 bases, and there are roughly about 25,000. Some of them are small, some of them are large, but that 10 to 15K is the average length. Now this information gets copied at each cell division. Just like all of us know, when we do cut and paste, we make errors. That's what cell does too. So sometimes these copies create errors. When an error happens, that cell, as I said, this is what encodes what the cell is supposed to do. If there is a copying error in copying what the cell is supposed to do, the child cell is going to do something slightly different from what it was supposed to do. When it does something slightly different, what happens? I mean, either the change that is made to the DNA is irrelevant, in which case it doesn't matter. Even if that copying error happened, that part of the gene was not controlling any function, so we won't even notice. Sometimes it's really bad. It's so bad that the cell is unable to survive. Let's say the part of the cell which metabolizes glucose, that part is destroyed. If that happens, the cell is not going to be able to consume glucose and get any energy. It's eventually going to die. So that's going to be flushed out of our system. So neither of these two is too bad. What becomes bad is if that slight change gives the cell a slight advantage over the other cells. Now, if that slight advantage in terms of survival, and that can happen in multiple ways. Either it starts growing very fast, it starts metabolizing very fast, if it starts reproducing fast, or if it refuses to die. If any of these functions are altered in the favor of the cell's survival, then we have a problem. Because not only have we created a cell which is slightly more likely to live, this cell is going to replicate, and this is going to continue replicating. Thankfully, it happens very rarely. And it's both good and bad. The whole evolution is dependent on this mechanism. The whole evolution of species is dependent on this. So, sorry.
So, could you please repeat the reasons why that happens? I mean, you said that it happens very rarely, but what are the possible reasons for this?
Okay, the question is, it's a very good question. Why does this copying error happen? So you have to think about cell mechanisms not in terms of digital computer-like behavior. It's biochemical. So a strand of DNA, it's a sequence of very large number of proteins. Each protein, it's a sequence of very large number of alphabets. Each alphabet is a nucleic acid. Now, the way division happens is that DNA is two complementary copies. There's A, B, C, D here. Let's say, actually there are 4 alphabets, A, C, T, G, and there's so there's two complementary copies of the code in each DNA. It splits and then attracts other molecules to come and join. Now in this process, this is a biochemical process. Sometimes the way other molecules are present in that vicinity, binding happens in an incorrect way. Some part gets deleted and the molecule is still stable. If you come up with a stable molecule, despite retaining an exact copy, you have an alteration during the copy.
So it's not that easy to quantify. Why?
Some factors accelerate the reason for this miscopying. Some factors, for example, we know very well that nicotine consumption accelerates the likelihood of such mutations. So presence of certain carcinogens, what they do is they make it more likely for these errors to happen. So there are ways of quantifying that. It's impossible to prevent that altogether. We don't want to prevent that altogether because we know that we came about it because of this mutation process. Everything started with a single cellular organism billions and billions of years ago, but we are different. And so it has its benefits. We evolve, we learn, the whole genetic code that comes as a part of it, but what we need to make sure is that we are able to, diseases which are caused by this miscopying, we need some way of handling them. Can I move on?
Yeah, sure. Thank you.
So these cells are copied meticulously, but errors happen. When errors happen, there are some errors which are bad, some which are neutral, both of which are good because the bad errors will make the cell immediately die. the neutral errors won't matter, at least in the short term. We'll see why neutral errors are not as neutral as they appear to be. And some are actually good for the cell, bad for the organism, or they could be good for the cell and bad for the organism. And that's what, invariably, a single such mutation is not something that, our body is too sensitive to, because we have an enormous amount of redundancy built into our system. So as you can see in the previous pathways network that I was showing you, apoptosis has one path, but that's not the only path. There are many other signals that are also leading to cell self-destruction. So a single mutation, usually our bodies are able to handle pretty effectively. But what happens is soon we'll have another mutation and another mutation and another mutation. And some combination of these ends up becoming a malignant cell, what we call the cancer. So this combination of multiple mutations, which collectively lead to a poor likelihood of normal behavior, that is what leads to cancer. Now, I talked about a copying error. Let's think about how likely this copying error is. I told you about what the length is, but the likelihood of an error in a gene as during each division is about 1 in a million, 1 into 10 to the power minus 6. That doesn't sound too bad. I think there's a 1 in a million chance that a gene will mutate, and that too, a mutation is very likely going to be ineffective anyway. But think about it this way. We all started from a single cell, the egg cell, and my body today has roughly 30 trillion cells. So through my lifespan, and there are cells which are being created and they're dying on a regular basis. Through my lifespan, that original cell would have eventually undergone 10 to the power 16 mutations, which basically means that there's about, divisions, I'm sorry. My original cell would have gone through about 10 to the power 16 divisions, which basically means that every gene would actually mutate about 10 to the power 10 times through my lifespan. Now that sounds pretty bad, but what saves us is that most of these mutations end up being neutral, or they end up being so bad that the cell doesn't survive, and the healthy part of the cell eventually carries forth the function of that gene.
And you mentioned that a single mutation is not usually enough to cause something really bad to happen. I would tend to disagree with, like, for breast cancer, there's something called the BRCA2 gene, right?
Yeah.
So that's a single mutation.
That's a single mutation that makes you likely to get breast cancer. It's not guaranteed to give you breast cancer. Why do you become likely? So it increases the likelihood of you getting breast cancer about 10 times more than the rest of the population. But that doesn't mean that you're bound to get breast cancer. And that likelihood comes from the fact that you need to develop other destructions in your cell control mechanisms for it to really become breast cancer.
And certain factors, like you mentioned, the probability is very low. But there are certain factors, you mentioned environment, like carcinogens or smoking or whatever. The other would also be I think which is largely random, is if you genetically are predisposed, like one of your parents had a defective gene and they passed it on to you.
So that's not a mutation anymore. You already inherited that.
Okay.
So, for example, if my parent has a mutated gene, BRCA2 and BRCA1 are two breast cancer causing genes, and they can be inherited through generations. Some progeny will actually develop cancer and some will not. So yes, if you inherited certain mutations, then you're already susceptible to, you're not susceptible to mutations, you're more susceptible to develop that disease because you already have that mutation. So I'll need to speed up a little bit. So the reason, again, I'll repeat, cancer becomes challenging is because we don't have too many universal treatment options. As I talked about just in the previous slide, that a cancer comes into my body not because of one mutation, not because of one single change, but there's a vector of changes which is pretty large, and this vector differs from every two individuals who have cancer. Two people who come into a doctor's clinic with breast cancer, their breast cancer signature vector is always going to be very different. So what treatment options do we have if the underlying causes are different? We basically take the hatchet approach, where we say that we'll surgically remove that part of the body, we'll kill all the cells in that vicinity using radiation, or we'll do chemotherapy. Each of these options is essentially trying to kill all cells, healthy and unhealthy cells in that region. Now that's been the tool that we've been using for quite some time, but it has problems because there's no way to remove 100% of the cells. The cells are not localized in a very narrow area. They have slight spread, so you cannot remove them effectively. If you don't remove them effectively, then the remaining cells will start proliferating again, and they start taking place of the ones that you've removed. The recurrence happens. Of the 25,000 genes, there's about 1,500 to 2,000 which are known to play some role in cancer. Their altered behavior is known to play some role in cancer. So essentially, you're looking at a 2,000 wide vector where the numbers could be, you can fill any number of, you can fill this vector in many different ways. So, and this vector defines one disease. Two people who come in will have a different vector, and hence their signature is very different. That's why it's been particularly hard to find one targeted medicine which can take care of cancer. You cannot find one medicine which says, oh, TP53 is one of the genes which is a tumor suppressor. If that function is inhibited in a person, I'll give something that accelerates that behavior. TP53 happens to be one of the most prevalent genes which causes cancer. So that's actually an example where you can develop something targeted. But in general, the disease develops not because of a single gene or 2 genes or 5 genes, but a large number of genes collectively leading to misfiring of the signals. So this is why I believe that this is one of the diseases where the role for precision medicine is absolutely essential. So it's caused by disruption in signaling. It's unique to each patient. No 2 patients are alike, and similarly, no 2 cancer tissues are alike. We need to understand how communication within the cell is working at a quantitative level. We need to model it effectively. And then we need to figure out how to intervene in that malfunctioning network to set it right. Now, where have we seen this before? As computer scientists, we've seen these flow networks in multiple places. We've seen that in simple graphs. We've seen that in communication networks, or we've seen this in VLSI networks. In each of these places, we have built tools to simulate the behavior of that network. We have built tools to figure out what will happen if a disruption is caused in one of the signals, how the rest of the network is going to behave. Can we model cellular pathways in a similar way, and then start applying the techniques which have been built over time? And that's the premise with which my company started. So this is a picture from our cancer model, or our model of the cell. So inside the cell, we have lots of constituents which are different types of molecules. Each of these molecules are taking part in multiple reactions. When you chain these reactions together, where a molecule participates in something, produce something else, which participates in something, another reaction, and so on, this chain is called a signaling pathway, where the signal I showed you, where an external protein comes close to the cell, which tells the cell to die, something inside the cell binds to that, and as a result of that, triggers a signal. So this signaling mechanism is the way by which the cell controls all its behavior. And what we want to do is to model this signaling pathway in a way that can be used to predict its behavior. So let's think of a cell as a collection of these reactions, where each reaction has a bunch of participants and some defined kinetics. By kinetics, I mean a rate of change of these participants. And there's a very nice mathematical model called Michaelis-Menten model, which articulates how any enzyme-controlled reaction is going to behave. Most protein-to-protein interactions are enzyme-controlled. So here you have the green guys, which are the things which are going into the reaction. These are called substrates. And the red guys, which are being produced by the reaction, the products. There is an enzyme E, which is represented in dark blue, which is enabling this reaction. So the enzyme E binds to the substrates, produces something called ES, which then divides again to, the equation is appearing in a very poor, unreadable format. But E plus S combines to create a complex of ES, which then divides to create E plus P. So the enzyme E is facilitating the substrate S, and the substrate S is not a single molecule, it could be a vector of molecules. The enzyme E is enabling the substrate S to convert into product P. And by observing the rates of these reactions, you can come up with a differential equation which says the rate of production of P, dP by dt, is defined by certain constants and some function of it, substrate and enzyme concentrations. Now that's so good because now I have a way of taking that qualitative cell behavior and representing that mathematically. If I do this for all the reactions that are happening inside the cell, suddenly I have a pretty nice mathematical articulation of that cell. So that's what we've done. We have basically, all these circles are molecules, their parameters are their concentration, and the square box in the middle is the reaction. The square box consumes 1 and 2 as substrates and produces 3 and 4 as products. The modulator A and I, these are modulating the rate of this reaction. A is activating it and I is inhibiting it. So this is one single reaction in the cell, and there are tens of thousands, hundreds of thousands of these which are happening in parallel. Once you put it all together, you have a beautiful representation in terms of a flow network that can now be seen purely mathematically. I don't need to know what what each of these molecules is. I can start calculating behavior of the cell in terms of the concentration of these molecules, because I know that the concentration of one molecule results in the cell death. So as long as that concentration is maintained at a certain value, I know that the cell death behavior is going to be normal. So we've gone from the qualitative model to a quantitative model for the cell, which is basically a system of ordinary differential equations. The solution to this system of ordinary differential equations gives me the equilibrium state of the cell, which tells me what the concentration of each of the molecules is, at what rate the reactions are moving in the forward and backward direction. Even during the equilibrium state, the reactions keep happening. And it allows me to predict the cell behavior. So we built a solver and a simulator that takes this mathematical model and tells me what the cell is going to do. Now, so far I'm still talking about the normal behavior of a cell, where somebody studied a protein and said that this protein interacts with these other things and at this rate. And that's in the normal cell. What happens when there's a mutation? What happens when I introduce a drug molecule into the system? And the beauty of the model is that since I've represented all reactions mathematically, I know that what is modulating this reaction. So if one of the modulators is mutated in a way that its activity is inhibited, that means that this reaction will now happen more slowly. So whatever substrates it was consuming or products it is producing, that will change. I can mathematically plug in this mutation and start seeing what the effect of that mutation is going to be. That's the nutshell of our model, where I can overlay the mutations as well as the drugs and compute whether the end steady state for the cell is something that is healthy or unhealthy. Now, with a sufficiently complete listing of this, and I say sufficiently complete because it's never going to be complete, but with a sufficiently complete listing of these reactions, we'll eventually get to a state, or we are at a state where we are able to predict some types of cancer reasonably accurately. The goal is to eventually get to a point where not just cancer, but all these non-communicable diseases is something that we should be able to predict fairly accurately. So the solution we have built is, as a person walks into a doctor's clinic with a genomic aberration leading to a cancer, he's tested for genomic signature of the tissue. That genomic signature, the genetic mutations are read in by our solution, which basically creates an avatar of the patient. Instead of we take the healthy cell model, on top of that we inject all the mutations, and now I have a malignant cell which basically represents what's happening inside the patient's body. We use this to again compute the steady state, and then the interesting part is that we are now able to say that I'll take all the drugs known to mankind, and one by one I will compute what the outcome will be. Most of them will probably fail, but some of them will not. And that's what becomes a positive therapy for the person. So by doing this, I'm able to identify therapies. And the interesting part is that given that I'm not actually, the person is not ingesting these medicines, he doesn't need to be, I don't need to be careful. I can give him any random molecules whose behavior I understand, and I should be able to still see what's happening to the system. So what we actually do is we don't give medicines just one medicine at a time. We create combinatorial combinations of all medicines in all concentrations and throw it on the system. Most of them fail. But the small number of them that do survive, that's the interesting therapy that the person is responding to. At least our model is predicting that and wherever our modeling is accurate, we are able to predict that we'll be doing a decent job. So we create the patient avatar and combinatorially run that through all the possible medical combination or medicine combinations and see which ones are A+ and which ones are C-. So I'll show you a small case study, after which we'll take some quick questions because I'm being told that I have only 5 minutes to left. So in this case study, this is actually a real patient. This is the number of mutations that were seen in the cancer tissue. Now that number is not small. And this is not the complete list. This is only the genes which are relevant to cancer. All the genes which are irrelevant to cancer have already been pruned out from this. So this number still remains of the order of a couple of hundred. There's a couple of hundred genes which are behaving in a wrong way for this particular patient as measured by the genomic sequencing tools. Now, if I give this to a doctor, what is a doctor going to do with it? Can you intuitively tell me that which 50 of these are combining together to actually lead to cancer? It's impossible for a human mind to intuitively predict something like that, which is why genomic sequencing has been around for last several years. Last couple of years, even the price has become fairly tractable, of the order of about $1,000 to $2,000. However, most of these tests are done and the doctors are not able to make use of this data because they have no way to consume this data and intuitively take a decision. This is where I believe that we are lucky to be in the computing field because computationally we are able to do something with this. So we take that and through our network, we compute which of the starting aberrations lead to some signaling disruption towards genes or proteins which have something direct to do with cancer. So starting with the initial few hundred mutations that I showed you, we are able to abstract out the system and identify the pathways which are misbehaving. This is actually, there's about 6 or 7 such figures that we eventually produce. I've shown you only one of them, but still they are fairly intuitively consumable, which exactly identify what pathways are leading to the increase in NF-kappa B1 protein, or which is leading to mTOR protein, which is eventually directly linked to the proliferation of the cancer. So that's something that we are able to compute. And on the right side, I show you the pathways where FNTA, which was one of the original aberrations in the system, leads to NF-kappa B1, which is actually one of the contributors to the disease. Now, initially, in the previous list, we only saw what is on the left side, and those things are not directly linked to cancer. However, what's at the end of these paths, if I show that to a doctor, the doctor can immediately identify, oh, NF-kappa B1 is misbehaving, or this is misbehaving. These are very regular things that they see in patient after patient after patient. What we are able to do is to start with the initial aberrations and identify the actual hotspots which are causing the cancer. Now, the therapy we computed for this person included metformin and sorafenib. So when I asked if there are any medical professionals here, they would have immediately stood up right now, what are you talking about? Because metformin happens to be a medicine that is given to type A diabetic patients, type 2 diabetic patients. And sorafenib is a medication that is given for kidney cancer, liver cancer, or thyroid carcinoma, not for MDS, which this patient is about. This patient was a blood cancer patient, and this sorafenib is not a regularly practiced medicine for blood cancer. So immediately I would be challenged, on what basis are you talking about this? Because these, how can you give a diabetes drug to a, but that's what our system is predicting. So to convince that we are right, we are also able to generate a pathway which tells me at what point is this drug acting so that the final cancer parameters are correctly tuned back to their normal state. So that's the system we have built, and we've got fairly decent results in a couple of types of cancers. There's a lot more work to do for continuing this to cover all cancers and then eventually all non-communicable diseases. We are able to expose what is causing the cancer. We are able to discover some novel therapies that would not have been attempted otherwise. In many cases, we've gotten extremely remarkable results for patients which had kind of gotten through all possible known treatments and not responded. Obviously, we have not succeeded everywhere. It's still work in progress, but I see a lot of opportunity in this space. And the reason it heartens me is because people like us, people coming from mathematics and software engineering and computing background, can now start playing a role in this piece. Thank you very much. I'll be happy to take some questions if I'm allowed to. Okay, 2 questions, I'm told.
One
Thank you.
question. In the case study that you showed, the person got treated?
So
He was cured?
So this is a case study that is still ongoing. This is a patient for which we have generated the recommendation. It's a very recent patient. The response is typically known after 3 months. The initial response could be positive, but that can turn around in a negative way. So it's too early to predict whether the person will respond or not.
Okay, and do you have it in India also? In India,
We are currently in the stage of clinical trials, and these clinical trials are being run in Florida, not in India.
Okay.
We are in talks with some hospitals in India. And eventually we would like to do it here as well, but we first need to, given our bandwidth, we need to limit our focus.
Okay, thank you very much.
Hi, Pranay.
Another question somewhere there?
Just last question.
Hi. Love the talk.
Thank you.
2 quick questions. One is, so are these just recommendations, or does CellWorks need to be FDA approved and I'll ask the second question also now. So so far it's mostly been radiation therapy and chemotherapy. So there is a new technology called CRISPR-Cas9, which essentially targets the root cause by editing DNA. And there have been an attempt in China to do it in a human being. So I would love to hear your thoughts on that.
Certainly. Let me also take the question from there and I'll try to combine the answer because this is the last question. Can you?
How to simulate the signal pathways inside.
Could you please use the mic?
Yeah. So what I was asking is about the scale of the simulation. So what you have shown us is the signal pathways inside a cell, right? But the cells do signal each other,
yes,
right? So the network is perhaps far larger and far densely connected.
Absolutely.
So that was the first part. And the second was, so there is some amount of stochasticity in it, right? So, I mean, it's not just a collection of ODEs, right? So there are probabilities involved.
Yes.
So determining the final equilibrium, etc., sounds tougher than how it looks.
Perfect. Very good questions. Both questions are very good and there'll be very different answers. So let me start one by one. So yes, there are many therapies that are being attempted, many ways of attempting therapy. And immunotherapy is one of them, and what you said, DNA editing is yet another. DNA editing requires us to understand where the changes are and what changes are essentially critical for us to be able to make an amendment. That's where the complexity of this vector of 2,000 genes becomes important. If you tell me that I showed you the 300 genes or so that were mutated in this guy, are you going to edit all the 300 of them? Are you going to pick the 6 that you will edit? Because none of these processes are 100% accurate, so when you edit something, you make mistakes. So you don't want to edit all 300 of them and then eventually cause something really bad. So understanding this disease is something that I believe is absolutely essential for any of these methods to work. Right? What was your first question again? Can you just? Does CellWorks need FDA approval? Absolutely, we do if we want to prescribe. And we are in the process of going through that. We will be going through that, and the clinical trial we are running is actually with an eye towards FDA
right
approval.
now, do you just simply give recommendations to doctors or oncologists?
So even our clinical trial has to be approved by FDA, that you can actually use this clinical trial and the outcome of this software recommendation for treating anything with human beings. So that goes through all that process. But the actual FDA approval to use it in production cases is a much longer process. Now, the scale of these computations, the scale of computation is huge. So we run our computation both on an in-house grid of computers as well as AWS cloud. So typically, it's not a single simulation that we do to find an answer for an individual. If they think about how many drugs we know of, we know about several hundred drugs. And if you generate 2 or 3 combinations of these several hundred drugs, we are talking about millions of potential combinations. We don't run millions of simulations. We prune down that intelligently and typically run a few thousand simulations for each person just to test out the drug. And each simulation is a fairly, I think on a typical i7 type of a computer, it takes about 5 to 10 minutes for one simulation. So that's the scale we are talking about. And you're absolutely right that the signaling is not just internal. There's a lot of external signals that feed in. And there's a lot more. The liver cell looks very different from the cardiac cell from another cell and so on. You're absolutely right about that. Cancer happens to be largely an intra-cell disease. So we can make a lot of progress for cancer just looking within the cell. That doesn't mean that we should not be looking outside, that we have to. We have to look at the microenvironment around each cell. We have to model that. We have to model each organ differently. And a lot of this work is, I'm not presenting something that's a concluded work. But what I'm saying is that with this work, we are able to model at least 3 types of blood cancers, AML, MDS, and MM. These 3 types of cancers in a fairly accurate way. And that's what the clinical trials have been showing so far. Thank you very much. You've been very patient. And I hope you like some aspects of using computing in biology.
Thank you so much. And this one's for you. Thank you so much.